Here's the final video that I'm going to make about Chapter 6. There are several components to learn about in Chapter 6 in the textbook. I'm trying to highlight them and build some stories around some of them, but not everything in Chapter 6. And this one is going to be about macrophage. So we talked about that in the previous video. That's a type of white blood cell that phagocytizes infecting bacteria. And macrophages can move. So this is going to be about cell migration. And in talking about cell migration, we're going to focus on the cytoskeleton. And so here's a figure from the textbook. And what I've done is I've said, okay, so take a look at how can we simplify this information. So the first thing is we've got these three columns, and so we've got these three components to something called the cytoskeleton. And the cytoskeleton, you can see down here visually, is a sort of structural framework for the cell that helps in a variety of different ways provide structure to the cell. And there are proteins that make up the cytoskeleton. So another example of proteins, we meet more proteins, and so here we've got three different kinds, and they are called microtubules, microfilaments, and intermediate filaments. And we're going to especially talk about microfilaments and microtubules. Those kind of difficult words to distinguish when you just learn them. The way I remember them is that I always say actin microfilaments, because microfilaments are made of actin, and microtubules are made of tubulin. So we got actin microfilaments and microtubules, and then it'd be great if we could learn a little bit about what each of these do. And intermediate filaments help keep the cell shape. So here's the nucleus, and intermediate filaments give the nucleus its spherical shape. So boom, we're done with intermediate filaments. If you could learn that, that'd be great. Intermediate filaments are made up of a variety of different proteins, and they help give the cell its shape, and shaped like the nucleus. And they're made up of a long polymer. This strand is a long polymer of building blocks, proteins, as is each of these. Then let's go to this one. Actin helps the cell change its shape. Here right below it, one way of changing its shape is muscle contraction. And so let's just skip forward to a quick view of muscle cell contraction. And you might already have heard the name actin, because actin and myosin are the big players in a muscle cell contraction. So that's an example of cell shape changing, and that's a good thing to learn about what microfilaments do. Actin microfilaments help the cell shape change, for example, in specialized examples like actin, like in muscle contraction. And then here are microtubules. They're made up of tubulin, and what do microtubules do? They help organelles move around, like vesicles. So all the time we've been talking about vesicles, it turns out that vesicles are moved along the train tracks of microtubules. So here's a vesicle heading toward the cell membrane to go through exocytosis. Here's a vesicle being pulled in from the cell membrane, going through endocytosis. And once exocytosis, but before exocytosis is accomplished, this vesicle has to get near the membrane, and there are little protein motors that bind to the microtubules and drag this vesicle to the membrane. Okay, so that's just a good look at that, and now we're going to focus in on a couple of stories about actin and actin microfilaments and microtubules, and whoops, they fit in here. So we talked about macrophages, and macrophages are white blood cells. There are a variety of different types of white blood cells that are important in the immune system, and macrophages are going to engulf through phagocytosis infecting bacteria. So macrophages fight bacterial infections, boom, like that. Sometimes the infection is out here, and the macrophage will crawl between the cells of the cell wall of the blood vessel and crawl out here and find those bacteria and phagocytize them. So that's what we're talking about. And now we're going to just talk about this macrophage moving. And as it's moving, it's going to change shape and it's going to move stuff in it. So now we're going to zoom in on a macrophage. And here's a macrophage moving, and it's moving that direction. How's it moving? Well, it has these various organelles and stuff inside of it. And back here, in the back of the cell, are actin microfilaments and myosin protein, and this is not a muscle cell, but these proteins can slide past each other using energy. It requires energy for movement, and as they do, they push the cell forward like somebody would describe a tube of toothpaste, where you can move the toothpaste up by squeezing the bottom of the tube. So one way the macrophage is moving toward those bacteria is it's pushing the contents of the cell forward. And that's going to take energy. This is the kind of work. It takes energy to contract your muscles. It takes that same energy to contract this mesh of actin microfilaments. And it's going to take the energy that's provided by a molecule that we're going to start talking about next, ATP. So now we're starting to see the dynamic cell requires energy. All of these different things we talked about requires energy to get done. And we're burning calories, and we're burning calories for a variety of reasons, and one of them is because our cells are doing real work. And here's a good example of that. So there's going to take ATP, and ATP is going to be catalyzed to break down to ADP, and in doing so, it's going to release energy to drive this forward. So that's one way the cell's moving. The other way the cell is moving is up here, and up here, the actin microfilaments, which are polymers, are being polymerized. So the actin microfilaments are growing, and that also takes ATP, and the growth of those microfilaments pushes this part out. So the cell is pushing itself forward, and it's also reaching forward, like your arm reaching forward to grab something. And it does grab something because this is the cell membrane pushing out, and there are membrane proteins, cell membrane proteins, that maybe we'll draw them a little bit like this, that are actually like hands that can reach forward and grab onto stuff. Then the cell will be pushed and pulled forward as it holds on and gets a physical grip. All right, and these proteins are called integrins. So we have another protein that we learn about, integrins, and we could look in detail about their different amino acid sequence and how it folds up to form these handholds. And what's going on here is this ATP will cause the polymerization of actin, and the hands will reach out, and if they find something to grab onto, they'll hold on, and the cell will be pulled forward and pushed forward and move in that direction. And so that's the source of energy. That's the use of energy to pull the cell forward, move the cell forward. What are these things grabbing onto? This is described, it's a little bit of a side note, this is described in the chapter 6 on the cell. These handholds are typically on the extracellular matrix. And the extracellular matrix, one example of that is cartilage. So cartilage is not cells, it's extracellular material like collagen and other materials that help strengthen tissues. So, your skin doesn't just fall apart because it's held together by this extracellular matrix, abbreviated ECM. And so the integrins are reaching forward and grabbing hold of the ECM because the ECM is kind of everywhere between cells, and so there's material to grab onto and pull forward. Okay, so let's say it grabs hold of the ECM, it then holds on. So these integrins are already holding on to the extracellular matrix, and there are integrins back here holding on to the extracellular matrix. Like that. So the next thing we're going to talk about is just to follow these integrins, these handholds, because what happens is they get a hold here, they hold on here, and then let go back here at the back of the cell. and they let go, and then look at what happened is we've got hands up here, but now they got pulled back here, and we need to get them back to the front of the cell. So as the cell moves forward through these forces, there's a recycling process that's happening inside the cell that is going to involve endocytosis back at the back of these hands. After they let go, they get endocytosed, and remember that means bend it in, form a coated vesicle, uncoat the vesicle, and now you've captured these hands in a vesicle. And what can you do? You can take them on train tracks of microtubules, right? And that's going to take some energy. That's going to take ATP. There's going to be a little molecular motor that's going to bind and pull this along, pull this vesicle along. And where is it taking it? It's taking it to the front of the cell, and it's bringing the handholds, recycling handholds, to the front of the cell and delivering them there. How? By exocytosis of this vesicle in order to get the hands, fresh hands, out to the front to grab hold. And you see, it's almost like the tread, you know, like on a tank or like a bulldozer, right? Where you've got the handholds and they're being pulled back like this, like that, and then you get recycled and brought up to the front again using microtubules. And so we got the cell moving, and it's moving through changes in the cytoskeleton, and a couple of really nice examples where the actin microfilaments are changing the cell shape. The overall cell shape is changing because we got polymerization happening out here, changing the shape of the cell out here, and then contraction happening back here, changing the cell shape, pushing stuff forward here. Then microtubules are moving stuff around inside the cell like vesicles, bringing those vesicles to the front of the cell, boom, like that. And so that's going to be our example where we learned some about the cytoskeleton and put a picture on the cytoskeleton. There's a lot more detail in the table of the cytoskeleton, but if you can simplify it and pick out those and use this as an example, you'll be in great shape.